phospholipid bilayer of vesicles, and within those vesicles, inside the hollow space of a vesicle, you have neurotransmitters. So neurotransmitters can be large, like they can be big proteins, or they can be small, like the size of an amino acid, or they could be as small as just a couple atoms big. So it's just, a neurotransmitter is just an umbrella term for anything that's going to be spat out of the axon terminal and interact with the next neuron. You had a question or no? Yeah.
Are there different types of neurotransmitters?
Oh yeah, there's neurotransmitters that make you feel happy, sad, angry, hungry, sleepy, frustrated.
Do we need to know the difference?
No. No.
Wait, so are they just like stored down there?
Yeah, they're just hanging out, waiting, ready to go.
What are the things that like strong?
Yeah, so the way that a vesicle gets to the end of a neuron is the inside of a neuron is filled with microtubules. It's filled with a cytoskeleton. Now, I normally don't draw those, and you normally don't see those in a textbook because it would just make the image look even more confusing. But these vesicles are in the cytoplasm, and there are little motor proteins that look like little legs that walk the vesicles down.
Those are the ones where like ATP like powers them down.
Yeah, exactly. So the way that they walk down is by hydrolyzing ATP, and eventually they reach the end of the axon terminal, and there they wait for an action potential.
Okay.
So remember, an action potential is going to start way over here at the axon hillock. If we get to -50, we get firing, then firing, then firing, then firing. We have sodium flooding in here. Some of that, and so you're going to have an action potential here. Some of that sodium is going to diffuse to the next voltage-gated sodium channels and make this area -50. You're going to have firing. This area gets to -50, you have firing. So it's a chain reaction that's going all the way down the length of the neuron. And eventually what happens is that when this region reaches -50, you have calcium voltage-gated ion channels. So now we're talking about sodium all the way down here, but at the very end you have calcium voltage-gated ion channels. When those calcium voltage-gated ion channels detect -50, they open up. Now instead of sodium flooding in, calcium is going to flood in, and that calcium flooding in is going to trigger these vesicles to do exocytosis. So when these vesicles do exocytosis, they spit out all of their neurotransmitters into the space between this neuron and the next neuron.
Which is a synapse.
Which is the synaptic cleft, yeah, or the synapse, yep. The space between.
Sodium goes in, and what goes in?
Calcium.
Okay.
So when these neurotransmitters diffuse into the synaptic cleft, they're going to interact with another neuron. So here's another neuron. Whoops. This is the dendrite of another neuron. So this is the presynaptic neuron. This is the postsynaptic neuron. The postsynaptic neuron has ion proteins, ion channel proteins in its plasma membrane. These proteins will bind to the neurotransmitters, or I guess the neurotransmitters will bind to the proteins, and those proteins will open up. So these are gated proteins that don't open because of a stimulus like sound or light or touch or heat. They're only opening in the presence of these neurotransmitters. If enough neurotransmitters are present to cause, let's pretend that this is a sodium-gated channel that's sensitive to these neurotransmitters. If they open and enough sodium starts diffusing in, and this region gets to -50, what's going to happen? you're gonna have, this neuron is now gonna fire. So the message goes from electrical in the form of an action potential to chemical, back to electrical. If, let's pretend instead of these being sodium channels, they are potassium channels. If the neurotransmitters bind to the potassium channels and the potassium channels open, what's gonna happen?
Just gonna go like that.
Potassium is gonna leave, which is gonna cause the inside to go from -70 to something like, I don't know, maybe -80. So in other words, neurotransmitters can sometimes cause the next cell to fire, or they can sometimes prevent the neuron from firing by hyperpolarizing it. So neurotransmitters both tell cells when to fire and when to be quiet and not fire.
So does that depend on what proteins are on the dendrite?
Yep, that depends on which proteins, it depends on which neurotransmitters are secreted, and it depends on which proteins are on the dendrite. That's gonna determine what the next neuron does.
So are neurotransmitters like proteins?
They can be proteins, they can be amino acids, they can be small organic molecules that aren't classified as proteins. They can be just a few atoms big, like nitrous oxide, NO2, is considered a neurotransmitter. So yeah, they're a bunch of different things. But the one thing that they all have in common is they all interact with neurons and cause neurons to either fire or not fire.
Are these types like randomly distributed through like neurons?
No, different parts of the brain and different cells within those parts of the brain, their job is to produce different types of neurotransmitters.